So now, sorry, I'm looking at the computer. Now I am doing the isolation of your own DNA. Hopefully your lab manuals look prettier than mine. This starts on page 66. And this is a really simple process. I'll even post a lab to the class website for you guys can do this at home technically. You don't have to have all the fancy stuff I have here. You could do it on your own. I have even done it at home before and it works beautifully. And there's actually a few ways you can do it at home, so maybe I'll post a couple different methods. But what I'm going to do is point out what all the different ingredients we're working with are here. We have this, what's called a hypertonic solution. This is a 0.9% saline solution. Kind of like Gatorade, it's salty, that's what it tastes like, and I put 10 milliliters of it into what's called a conical tube. You can see there's little markings on it, so you can see how much is inside. We've got, it says a 10 milliliter graduated cylinder. This is going to be for measuring out this detergent protease mixture. And then eventually we'll add this ethanol. Okay, so the way it works is, I'll see if I can get my assistant to come over and do this with me. We're going to start out by taking these.
Oh, is that me?
That's you. These we have two conicals, one for me, one for her. You can come over here. And we're going to literally, it's going to look like we're drinking it, but we're just going to put it in our mouth and swish it for 30 seconds. So this is step number 1 here. Take the lid off. Remember, you're not going to swallow it.
Wait, you're making me drink something?
No, you don't drink it. You stick it in your mouth, you swish it around, and then we're going to spit it back out in here. But you got to swish it for 30 seconds. Okay? You ready? It's not that bad. Don't do that. 1, 2, 3, go.
So much braver
Okay.
than all of you.
Okay, so now we've done that part. And the next thing we're going to do, she makes it look a lot more dramatic than it is, but the next thing is we're going to add 5, I'm on step 2 on page 66 now, add 5 milliliters of the detergent protease solution to the tube. So I'm going to go get your water bottle and wash out your mouth, goofball. So here's the detergent protease. I'm going to measure out 5 milliliters. Ooh, it's thick. I think usually we do this with a, what is it called? A little tube, a different type of a, what's called? Pipette. We usually do measure this out with a pipette, but we're going to do it this way this time. Okay. And once we get this added, the directions are to put the lid on the tube and gently mix by rocking the tube for 2 to 3 minutes. So this will be a not terribly interesting part to watch, but I'll show you how it starts. Okay, you ready? We have to rock our tubes back and forth, Rosie.
Oh. What do you mean?
So we have to stand here and for about 2 minutes. I'll show you. We've got 2 minutes on here. we're just going to go like this. Just rock. So now we've mixed the salt water plus our spit with soap
Ew.
and this stuff called protease. And you can see it just looks like,
Why don't we just violently shake
okay, that's a good question. Why don't we just violently shake it? There's a couple
Like-
reasons. If you violently shake it, it's going to get, you can already see there's a lot of bubbles in there. It'll get really, really, really foamy, which is not helpful. And the other reason is all we're trying to do now is what's called lyse, or split open the cells that were in our mouth and now they're in here. We want to split them open gently so that we can release the DNA, as it were, from the inside of the cells. So that's why we treat it like a baby. Ooh, okay, why didn't we do our blood? Because blood is technically a biohazard, and then we'd have to dispose of it super duper carefully.
Oh.
But people spit all the time, so I guess we're hoping you don't
What about
have any.
skin cells? Like, you know.
That is, the inside of your mouth is skin cells.
What about hair?
That's much harder to get DNA from, because really you can only extract the DNA from the root where it grows. The rest of your hair is technically just keratin and
That would've
doesn't
been
have
so
DNA in it. Huh?
I said blood would have been so cool.
Blood would have been so cool.
Not that I think like drinking blood is cool. It's not. Don't drink blood, please.
Don't drink blood. I mean, unless you're starving or something weird.
Don't drink blood, period. We don't resort to cannibalism.
There you go. All right, so now we're going to do 10 milliliters of ethanol. In the lab manual, okay, and this is step number 4 on page 66. In the lab manual, they say ice cold. Technically, it doesn't, the temperature of the ethanol matters very, very little for this portion of the experiment. So don't get caught up in the temperature. Here's my tube. We're going to film this and hope that you can watch the actual DNA come out of solution. So you'll see that it says, tilt the tube of the cell detergent solution and gently pour the ethanol down the side of the tube so that it forms a layer on top of this mixture. And do not mix it vigorously. So here's one of the cool little fun things you're going to see about this. You just watch. Can you see how there's like a layer forming? The ethanol is actually floating. Can you see that? So this up top here, that's the ethanol layer. And this down here is the mixture of the salt water and the detergent and the other stuff. Maybe you can see it. Can you see it better that way? And maybe, trying to figure out here, kind of, if I could put a background behind it. White background? No, you can't see anything that way. My shirt's almost a better background. Now you can really see. Here, this clear layer is the ethanol. This is what we call like the mixture. That's my spit plus the salt water plus everything. And if I get close, can you see those bubbles? So the bubbles are the DNA coming out of solution. If I gently sort of agitate it, those little bubbles, it almost looks like, I don't know how to describe it. To me, it almost looks like you're looking at the stars in the universe and suddenly they're coming into motion. So this is what it looks like when your DNA comes out of solution. That's the effect that the ethanol has on your mixture. Come back over to this side here. And that is my DNA, that clumpy, almost boogery looking stuff in there. Kind of cool, huh?